Hey everyone, today I'm going to teach you about staining of bacteria. So let's get started without wasting a time. Staining of bacteria. So first of all, you should all know that why there is need of staining of bacteria. Why we stain the bacteria? As we all know that they are microorganisms. Bacteria are semi-transparent and it is very difficult to see them without staining. सो स्टेन करने से हमें क्या क्या हेल्प मिलती है दैट मेक द बैक्टीरिया विजिबल एंड उसकी शेप और साइज़ हम स्टडी कर सकते हैं, उसका इंटरनल स्ट्रक्चर हम स्टडी कर सकते हैं ऑन स्पेशल स्टेनिंग and it also help in identification and classification of bacteria into different groups. Next, let's discuss about stain and dye. We can't use any dye to stain the bacteria. Dyes are the coloring agent used for general purpose, as in textile industries that color your clothes, na? And stains, they are coloring agent manufactured under specification. Those are specifically designed for staining the microorganisms or biological material. Next is example of stains are methylene blue, crystal violet, safranin, eosin and nigrosin. I'm telling just these examples because these are used majorly in microbiological experiment. Otherwise some other stains are also available in the market. Now let's discuss about the types of staining. Firstly, simple staining, then differential staining, negative staining and special staining. In simple staining, we use only one stain to stain a particular, or you can say one of the bacteria and study them under the microscope. Next is differential staining, in which we gonna use two stains and two of the bacteria, where we are trying to differentiate between two of the bacteria. We're going to use differential staining, in which we are using two stains also. Then next is negative staining. In negative staining, there is a black background and the colorless kind of microorganism will appear, just like the all-time photographs. In special staining, there is demonstration of internal structure of bacteria, any particular internal structure. We're gonna use specific stain for specific structure of the bacteria to visualize under the microscope and study them. Firstly, let's discuss about differential staining. As we all know, differential staining is used to differentiate between two bacteria. There are three kinds of differential stainings: Gram staining, acid-fast staining and modified Ziehl-Neelsen staining. Acid-fast staining is also known as Ziehl-Neelsen staining. Gram staining, it divides the bacteria into two groups, gram positive and gram negative, which we widely use also. It was given by Hans Christian Gram and it is based on the difference in cell wall composition of two bacteria. First of all, you should know the principle of gram staining. It is that because of the difference in the cell wall composition of the bacteria, they're gonna take different stains. In the procedure of staining, first of all, you have to take inocula of bacteria from culture and bacteria with the help of inoculation loop on glass slide. Then fix the smear at 37 degrees Celsius by using, you can use the heat also. Then add the crystal violet, that is primary stain, onto the slide for 1 to 2 minutes. Then wash it. Then add the iodine, which is acting as a moderant, for 1 to 2 minutes on the slide. Why iodine is called as moderant? Because it helps crystal violet to penetrate deep and form the complex in the cell wall. Okay, next add the decolorizer for 15 to 20 seconds. Then add the safranin, that is counter stain, for 30 to 40 seconds. Then wash and air dry the slide. And next, the result is going to be either gram positive or gram negative bacteria. If we are viewing pink colored colonies under the microscope, then they definitely gonna be the gram negative bacteria. And if you are viewing blue colored colonies under the microscope, they will be gram positive bacteria. If they are lying in the chain form, so they are called streptococcus in case of round shape and streptobacilli in case of rod shaped. Otherwise, if they are lying in the bunches, they are called staphylococcus on round shape and staphylobacilli on rod shape. I'm going to show you slide next. You have to differentiate between two of them. So, which one is the gram positive and gram negative bacteria? Yeah, left one is the gram positive bacteria and right one is the gram negative bacteria. Why it is so? Because it is stained blue and it is stained pink under the microscope visibility. Okay, I hope you understand better. Thank you. Have a good time.